okay, that was intended to measure the connectivity map. So measure in some way or another, where is the path of a molecule when you are looking at many molecules inside the cell, not only one. And this is inspired and developed on the base of the pair correlation. In that case, instead of the regular pair correlation that Francesco Palomba introduced today, the 2D-pair correlation. And we will learn how, using some algorithms in order to understand how is deformated the pair correlation function in the 2D, we are capable of tracing maps of the molecular connectivity inside the cell. So the main goal of this approach was to solve this issue. And the issue is, well, if you have a single molecule, it's quite easy to track. Able to see what is the path of this molecule here and where it's going when it need to get, I don't know, Coke or something in the medium of this crowd. But the concept that we want to take here is, instead of thinking all these people as a single, we can make measurement in order to see how is the flow of molecule, right, on an average sense. A bar, many molecules will go to this bar, and at the end, we will see basically a path that all the molecules go to the bar, or all the molecules go to the bathroom, or something like that. So it's a statistical analysis of many molecules doing the same that we will obtain the information in order to create a path. And I will try to convince how to do it. So Francesco already introduced all these concepts, so I will go very fast. So originally, we discussed about how important was the FCS, and with the FCS, we are in a point, and the molecules are passing through the volume, and we analyze this with the autocorrelation function, and from the autocorrelation function, we can obtain the diffusion, as well as the number of molecules. Also, Francesco tell us about this double foci project that was developed by Jonas Reis and Petrashvili, in which you can see an amplitude in the first foci and then an amplitude in the other foci, and by cross-correlating the two, you can tell that this was the same molecule. And Francesco also stressed the concept, the very important concept, these are the same molecule. It's not another molecule that takes place in one place, in one of the foci or the other, it's the same molecule. And he introduced the pair correlation function, which is basically the fluctuation and time 0, position 0, and time plus tau and position delta R here, right, normalized by the two. And he explained how important it is to figure it out if you have a barrier of an oscillatory. And I will explain a little bit more this in the next slide. Just to remind, what we are talking is, well, if you have a molecule coming from here, right, from 0, if you want to go and see if you will catch this molecule at a certain distance, you see by the probability of finding the molecule that, well, if you spend the time, and you see here tau, if you spend a time, the molecule will get there. Of course, because you have many molecules going in many directions, you get only a fraction of the original molecules that autocorrelate at the time 0. Right? So only, I don't know, 1/10 or even less molecule will get at this point that we are exploring. But still, you will have a statistic, and this is the purpose of this. And then we learned that basically, if we trace pair correlation at this different distance, we will see this is the kind of Carper that you collect over time, and then the autocorrelation function at time 0, you see the maximum amplitude is in 0, and then it's decay. And here you see that the maximum is not in 0, it is a little bit farther, it's this cyan line here, is because it's taking time to get there, right? And this is the reason for why you get the amplitude here. And also we learned that, if the diffusion is different, you will get farther or closer depending on the constant diffusion of the molecule. And this is also important for the purpose of this two-pair correlation. But to illustrate this is the situation. We have a molecule, and the molecule go from one to another, and if it takes some time, the maximum will appear in the time scale at some point here. If there is a barrier, the molecule will never get there, and there will be a very sharp decay because there is no possibility to get to the other side, right? To the other point where we are taking the image. If there is an obstacle, it will take far longer time, so we will expect to see the maximum of the pair correlation function a little bit farther in time. So these are the basically mainly three situations we may find. And it's interesting to stress and to understand that this approach is intended not only to see the diffusion coefficients, is a little bit farther, is to understand what are the opportunity to find objects that you cannot even see, right? Because you don't know in the cell if there is an organelle or if there is a liquid phase or if there is a barrier by, membrane or whatever. So with this approach, you can highlight this because you will lack a connectivity, or you will lack a pair correlation amplitude over time. So, and this was very well explained by Enrico, how powerful can be in the analysis of molecules going in and out of the nucleus, and I don't want to spend time in this. But when I joined the LFD, Enrico was already working on this idea of do the 2D pair correlation, and this was inspired on the MRI, brain connectivity, where you can see what are the connection of neurons by, seeing the connectivity of the water. So let me explain the concept. The concept is the following. If we have now, instead of a point, and we scan in a line, we have an array that detect all the pixels at the same time, very, very fast between stack and stack, we can do a pair correlation between all the points. So this point with all the distance at this radio. But this is true not only for this pixel, but all the pixels in this image. And how we can do that? Well, with a camera, right? With a camera, we can collect very, very quick in terms of millisecond time scale, right? 5, 10 millisecond time scale. All the pixels, so millions of pixels, 2048 by 2048 in the new CMOS cameras at this rate. And if we collect enough number of stacks, 10,000, we can do this 2D pair correlation for all the possible position for all the possible pixels in the image. As you can imagine, this is a colossal amount of calculation, right? But, well, if you have a good programmer, it will manage, I will make, and you can immediately see that if there is any obstacle in one direction or another, the pair correlation will be deformed, will change the shape. If you have an anisotropic diffusion, which means that in all directions you have the same probability of finding the molecule over certain distance, over certain time, you will have a pattern like this for the pair correlation function in this pixel. However, if you have a barrier, you will see that there is a heterogeneous distribution. In particular, one direction, the pair correlation will be farther than the other because the diffusion in this direction is, is possible, and in the other is not. How we can analyze this information? Well, in principle, we can take some algorithms in order to measure the first and second moment of this distribution, which is basically another thing that the long and the short axis of this distribution, pixel by pixel. So what we do first is we calculate the pair correlation function for all the pixels in the image. We save an image. This is an image of 32 by 32 pixels. And then we analyze one by one all these images. And we are using this analysis, we assign a calculation that I will explain in a minute. The calculation that we did is how the format is this, how the form is this 2D pair correlation function, and this is analyzed as the eccentricity or anisotropy. And I will show, I think I will show the calculation here, but basically is long is one respect to the other, and anisotropy is one way, and the eccentricity is another way to do it. But if you think conceptually, we have like a pipe here, right? A tube where the molecule cannot get there. You will see, connectivity inside. And if you have a barrier from the exterior, you will see, you know, obstacles here. And what we define is a dredge hole where if the value in this pixel, and this big stick shows what is the preferential diffusion. So, right, this is the concept of this. Well, first of all, we need always to test this with simulation, and this is a simulation that was run with the software. This is a pipe, like a little tube. This is all filled with the same concentration of molecule, and we leave them to diffuse very free. And then we measure the part correlation after 10,000 stack or more. What is the anisotropy, the 2D part correlation function in different places. So let's begin with the point one. The point one is a pixel right in the wall of this simulation. And you see that there is no correlation to this side, of course, because there is nothing outside. And all the other directions are, you know, equal probable. So in that case, we will draw in this pixel a line in this direction, right? If we are in a point like 2, where we are with all probability of diffusion in all direction, we get basically an image like this, which is all direction equal. If we are inside of this pipe, you see that the probability is only to go in one direction or another, and we will draw a line in that thing. So we build an eccentricity calculation, which is how you know, directed is the diffusion, and you see that in the walls and inside this tube, the eccentricity is very high. And if we draw a line to select only the pixel above certain threshold, we will have only the connectivity map along the pipe, as you can see here. And this is the whole concept of the thing. So we will be drawing basically connectivity maps in such a way where we can see the behavior of many molecules going on that sense. Then with the more simulation, we simulate like a vesicles here, and you see that they are going around. It's very sensitive, and you can see pixel by pixel, even if they are changing in the angle. You can see when you have corners that you can see also the change in angle. You can do even a mesh like this one, or you can do crazy thing when you get bored and you start to play around. But then you need to test with truly samples. And these samples are groups that we fill with fluorescing, and the fluorescing will free diffuse along the axis. We collect the stacks, and then we do the eccentricity, and then we do the connectivity map. And you see that the direction of the connectivity is along these things. So the molecule cannot go in other direction. You can only see connectivity along this pipe. But this is quite simple solution. There is no other chance that the molecule go around, right? There is no curve in this pipe. So we decided to stress the method using a biological system, not in vivo, but still biological. And this is a giant unimanular vesicles. The giant unimanular vesicles is a vesicle with one single bilayer. And this bilayer has basically a dye, this dye, C16. And basically, if you think about this molecule will be able only to diffuse surrounding this ring, because it's where it is fluorescent. There is no chance that the dye, which is a lipid, can get in or out of the membrane. So this is a complex system, because it will need to capture if there are diffusion in some way, always at the tangent at the point where you are, right? And this is exactly what will happen. And you can see here, this is the connectivity, and the connectivity is barely see here, but these are all sticks that show all the perimeter of the thing. There are a little bit of noise that you can see around, but basically it worked very, very well. And then came to the problem, what instrument we are going to do. And yesterday I told you that when we start this project, the problem was we were working with the upright microscope. The right microscope, we have a cell looking by the sagittal perspective. And it was very, very bad, because we barely can see something else more than the nucleus, right, of the cell. So we build the SPIM, and I want to show you here how it look like a set of data. Okay, I know. So this is usually a set of data. This is what we are looking. We are looking at this, basically a lot of noise, a little bit of fluorescence. And with this, we basically generate this kind of maps where we can start to see the connectivity, and you can see, I don't know, the barriers of the cell. You can see, you know, the interior of the nucleo, the nucleolus, etc., and some other connectivities in the cytoplasm. And here, sorry, here we can see much better. So this is just a picture to illustrate how is the configuration of the collection of the data. And you can immediately see here a map of eccentricity and some other stuff here. And I want to stop a little bit here because we calculate something which is very interesting. It's called the velocity, and the velocity is, well, when we measure this pair correlation, there is a center of mass of the pair correlation, and there is basically then a measurement of the center of the pair correlation. So the center of the pair correlation can be a semicircle. The center will be basically where is the radius equal in all directions. But the center of mass will be displaced because it's the whole set of data. Well, this shift in the between the center of the pair correlation and the center of mass of the pair correlation, it tells you if you have a barrier. Because if you don't have a barrier, the center of mass and the center of pair correlation will be the same. But if you have a strong barrier, it will shift. And this is a way that we find to analyze, and you can immediately start to see all these trabeculated structures inside the cytoplasm that we believe are, you know, ER and some other thing. I didn't say, but this is basically just green fluorescent protein free diffusing in the cell. There is no attached to anything. Green fluorescent protein is very nice because it's inert. It doesn't interact with anything inside the cell, so it's a perfect proof. But you can attach it to other proteins, and there are some works from, well, indeed, Julia shows some beautiful work with this channel, potassium channel, and Nick Hede has some other protein with MAHC molecules, etc. And it's very powerful technique. But so far, you need a special microscope, and only we have this size SPIM to do this fluctuation in a SPIM configuration. So we also wanted to do on a TIRF, and it's a perfect kind of analysis for TIRF, as Julia showed, because we can collect only information from what is near to the glass. And we acquire a lot of stack, and you can see that we can trace map, in this case, of the EGF receptor labeled with EGFP. So this is a epidermal growth factor receptor. And you see that the anisotropy and the anisotropy angle shows that there are preferential diffusion in one direction or another. Here, the angle means blue is vertical, red is horizontal, and you see that there are specific pattern. One point what is very interesting is if you collect at different distance, you get different maps because you get time and an opportunity to get farther, right? And this is also very valuable. And we analyze this comparison comparing the different anisotropy levels, like very high, which means there is only one direction, and also lower anisotropy, which means that eventually you have not that preferential diffusion. And we did analysis in order to compare with the long axis of the cell to try to identify, and this is possible. And the other important thing is we can analyze the different level of connectivities by the number of the anisotropy, and this enabled to analyze cluster of diffusion and to take advantage of taking advantage of imaging processing tools. We can also study the shape and the size and the distribution of these connectivities clusters that can be important in this case for the EGFR receptor. And all this basically give a lot of quantitative information about diffusion. But the really important point here, as Julia stresses, with the same set of data, you can acquire the connectivity maps. So you will have an idea of how, organized is the diffusion or the organized is the diffusion. But at the same time, you can calculate the IMSD, so the image mean square displacement, and you can, on top of the connectivity, you can put basically the law of diffusion of the molecule you are studying, and you can, have molecules that are diffusing as a Brownian, and you will get a line, or you have confinement, high confinement, low confinement, or active diffusion when it's positive, the curvature here. And you can also do some other analysis, like this introduced by Paul Wassman lab, which is called STIC, by spatiotemporal image correlation spectroscopy, in which if you look at the autocorrelation function over the time and you see that there is a displacement in one direction, you can trace the vector flow. So one point that I didn't say, but it's important to highlight is, in the anisotropy analysis, in the connectivity map, we know what is the preferential, but I cannot tell you if it's going in one direction or in the other. It's the majority of the diffusion in this line, right? But I cannot tell you if it's going up or down. With this approach, you can measure the vector flow, so you can see in which direction the diffusion is. So with all this technique, you can combine number and brightness, and nowadays Elizabeth Hein has developed an updated version of the PAR connectivity, in which, first of all, they select the fluctuation based on the amplitude, which means the brightness. So you can do the PAR correlation analysis of the oligomers or the monomers. So you can first analyze the molecules that are diffusing as a monomer, and you get a map, and then you analyze the molecules that are diffusing as a dimer, and you get a map, or a tetramer, or whatever. And this is very nice work, and it's really powerful too. But so far, in both cases, we have, first of all, an spin, and then we have a TIRF. So you need to have this kind of technology in your lab. When the ID detectors arrived to the market, I was in the LFD, and we ordered the ID detector not because of the use that ZEISS was interested to do. Well, first of all, Enrico wanted to use the ID detector as a camera, and he, with Lorenzo, developed a very nice approach where he introduced the PAR connectivity inside this camera. These are 32 channels in parallel, and this is a nano camera because each of this actuator is 200 nanometers. So you can do all this at super resolution, and they did a beautiful work on that. But the other approach, and this, well, by the way, the ID detector was intended to break the diffraction limit to the half, right? So we never use it for that, but rather we did this. And the other important thing, they claim to use the ID detector in a way that you can collect 4 lines at the same time instead of 1 line at a time. And this speed up the acquisition by 4. And this was a way of ZEISS to increase the frame rate to compete with the light sheet and with other camera-based instrument, but still with the really high signal to noise that you get with detectors. And this was particularly interesting for people who work with calcium and the spark of calcium, etc. But we took advantage of this because using this, we increased the scanning by 4 times, which means that we can decrease the scanning of a frame quite a lot. And finally, we were able to acquire a really decent field of view, like this one or this one, at the rate more or less of a camera, 20 milliseconds per frame with this approach, using a confocal microscope. And then we check if we can, and now the maps are really much more defined, and you can see a lot of more stuff inside the nucleus, inside the cytoplasma. And this was a paper that we published later on. And in particular, we wanted to stress this technique to study this new kind of organelle that take a lot of attention nowadays, the liquid phase separation. And basically, we pay our attention in the nucleolus, which is the oldest liquid phase separation we know. There is no barrier here, right? There is no membrane, but still we can see there are, some barrier for the diffusion of EGFP. And then if you wait enough, you can see that some of the barrier can break and some of the barrier get remains.